Hello, dear students. I hope everyone is fine and safe. Uh, I am Ashima Hassan, your TA for the second part of our course, Bio 2000. Uh, First of all, I would revise the previous quest. So for anyone who has any questions, so everything would be clear for you. So first of all, we have the true and false. Mitosis and meiosis division takes place in prokaryotes and eukaryotes for sure. It's false. It's only occur in the eukaryotes because actually eukaryotes are multicellular organisms. So their cells would undergo division and propagation, giving more than one cell, while prokaryotes are only unicellular organisms. In mitosis, after this stage, cells have 23 chromosomes. No, actually, in mitosis, cells would have 46 chromosomes because in mitosis, we are duplicating or not duplicating, we are preserving the number of chromosomes. We don't either increase or decrease, it's 46. If the cell would undergo intracytokinesis, it must enter the mitosis. The preparation for mitosis occur in the intraphase. Cell-specific proteins are produced at the interface because actually the cell, it's the first stage in which the cell need to prepare the proteins for its further division. Then this is a prophase. It's from another diagram, but actually this is a prophase before here. We have the centrioles division and the spindle fiber forming and the homologous chromosomes. They are still in a paired form. So this is a prophase. The chromosomes align on the equator of the cell at the metaphase. I guess most of you did that well. At the end of meiosis 1, the two daughter cells are formed and contain 46 because at meiosis 1 we still have diploid number of chromosomes, while in meiosis 2 the chromosomes or the cells have only the haploid number of chromosomes. Okay, so don't get confused. In mitosis we also have 46 chromosomes because we are not dividing the number of chromosomes, okay? While in meiosis 1, we still have 46, but in meiosis 2, they would be 23. This is the telophase. Lots of you also did that. This is a scanning electron microscopy. We could say that, yes, because actually, what is the meaning of mitosis? Mitosis meaning that the number of the cells undergo division by preserving the number of chromosomes. So in meiosis, first it would undergo mitosis, then probably cell chromosomes would be divided. Okay, the internal organ would undergo to expand their cells is for sure mitosis because we need to renew the damaged cells, increase the number of the cells, preserving the number of chromosomes through mitosis. So if I need to increase the number of the cells and preserve the number of chromosomes, so it would be mitosis. Klinefelter syndrome, for sure, it occurs due to any, what we can say, any misregulation at the meiosis, because actually here the number of chromosomes increase, meaning that it's occurring one of the gametes before getting crossed, one of them has extra chromosome. So for sure the defect would be at meiosis, any step in meiosis. I consider all those who telling me any step in the meiosis. Actually, it's in the interphase because it's a preparatory one, but as you said, meiosis, it would be fine to me. Good. Let's start our today. We have a very interesting topic about genome technology and DNA and RNA and what is meant by genetic engineering. First, we would talk about the properties of the DNA, and we would talk also about the genetic transformation of bacteria. Okay, and you know that all the living organisms would have either DNA or RNA. They are the biomacromolecules. DNA is a deoxyribonucleic acid, while the RNA is a ribonucleic acid. Most of prokaryotes have RNA. Actually, some prokaryotes also have DNA, but they are rare. Also, some viruses are DNA virus. For the deoxyribonucleic acids, they are found in only eukaryotes, multicellular organisms. For sure, you know that adenine would bind with thymine, cytosine with guanine. they are binding together by hydrogen bond, the A with T by 2 hydrogen bonds, the C cytosine with guanine with 3 hydrogen bonds. Then we have to say that this is a double helix structure. Here, this is a next nucleotide. This is the backbone of the DNA formed due to the phosphodiester bond, while the lateral bonds are hydrogen bond. Please keep attention for that. What makes DNA unique is the fact that one of the ATCG bases is attached to the C1 atoms at each sugar. You get the point? So forming the deoxyribonucleotide, that here the adenine or whatever the what we can say, the bases by adenine, guanine, cytosine, or thiamine would bind to carbon one of the ribose or deoxyribose sugar. Okay, so this is what makes DNA very unique, that one of the nitrogenous bases are would bind to the C1 atom of each sugar. In the RNA, it's a ribose phosphate sequence A, G, and U. Case of the RNA. The sugar phosphate backbone is a skeleton, as we mentioned, is a skeleton of the backbone of the DNA. The second strand, as in DNA molecule, is wound around so that the bases on the two strands are positioned according to the AT and GC rule. This would form, this is considered to be the double helix structure, or sometimes they call it Watson-Crick structure, which is not 100% correct nomenclature. Anyhow, okay, so the two strands in a DNA molecule, the covalently bound to the C1 atoms of the sugar phosphate backbone. So if we need to know what make them very stable, that the nitrogenous bases are covalently bound to the C1 atoms in the sugar phosphate backbone and coordinated with one another according to the Watson-Crick base pairing. Okay. It looked like a necklace. Beads came in 4 different colors, A, T, G, and C. Okay, so this is a single strand DNA. This is a 2 matched strands of DNA. This is a double helix. When talking about DNA, we usually talk about base pairs because the human genome comprises of 3 times 10 to the power 9 base pairs, and the 2 strands of the double strand DNA are identical. The double and triple lines between ATG bases in the 2 strands are non-covalent chemical bonds because they are formed by hydrogen bond. And for sure, hydrogen bond is what we can say is less stable than covalent bond. So when we perform DNA PCR or polymerase chain reaction, we have to break this hydrogen bonding. So the hydrogen bonding is a weak bond, and it could be break through increasing the temperature. So we would perform increase the temperature from 37 to 95 degrees Celsius degree. So we would break the hydrogen bonds and separate that into single double strands of DNA. This is called the denaturation process. Okay, so we would hit the DNA double strand that would result in the breakdown of the hydrogen bond. So the double strand DNA would be single strand DNA. Okay, so if we need to denaturate more than the hydrogen bond, and we need to break the bonds between carbon, nitrogen, oxygen, phosphorus, and hydrogen atoms, so we need to perform a chemical action or to use an enzyme. For instance, we can rely on the catalytic activity of the DNA, of the enzyme DNase 1, which promotes cleavage of oxygen phosphorus bonds in the DNA skeleton, which is a phosphodiester bond. So here, if I need to break that portion, I would use the enzyme DNase. If I need to break that portion, I need to use only the temperature, okay? So DNase would result in that shape, while heat denaturation would only cleave the two strands into single-stranded DNA. Okay, our experiments would rely on, we would use both the heat to denaturate the double strand and the DNA enzyme to split the phosphodiester bond. So we would study the heat-induced denaturation and the effect of cutting using the DNase. The trick here is to do it slowly without disturbing the two layers, because the bottom DNA solution and the top ethanol, at that time, the DNA is very fragile. So if I disturb the two layers, or I check the experiment vigorously, that might result in degradation of the DNA. If I need to analysis of the behavior of the denaturated DNA, I would make a gel to understand the behavior of the single-stranded DNA in comparison to double-stranded DNA. So the single-stranded DNA on the gel would run very fast, more than the double-stranded DNA. While in case of the effect of the DNase enzyme, it would result in formation of double-stranded DNA, but in small fragments. So at that moment, if I put the gel together with a control one, they would yield small fragments, different fragments, but they are not that fast in comparison to the single-stranded DNA. We need to do that homework. You have to find the properties of the double-stranded DNA, the appearance of the solution when shelled with ethanol. You need to complete that genetic alphabet, and this is a bonus one. Then we talk about the second part of our lab, would be about cloning, molecular cloning. The point that, as you know, that DNA is a basis of inheritance in lots of organisms. So, and it comes in two copies. The change in the DNA sequence would result in changing in the genes, and so the expressed proteins and the whole genome of the organism. In bacteria, genetic change can induce either great sensitivity or resistance to certain chemicals or antibiotics, which is a big problem that we are facing today, because actually lots of bacteria are showing resistance to antibiotics because people are used antibiotics in a very wrong way. And bacteria, although they are prokaryotes, but they are also very adaptive and very rooted to the surrounding environment. We will talk about ampicillin, which is one of the broad spectrum antibiotics with beta-lactam. You don't have to memorize that, you just need to know it. And it shows it shows a big effect on disturbing the bacterial cell wall, and thus leads to its death, most of type of bacteria. Some strains undergo mutation and are able to synthesize beta-lactamase in their cells. So How does those bacteria show resistance to the ampicillin antibiotics? They have beta lactamase in their cells, and it catalyzes and enhance the opening of a beta lactam ring, and thus prevents the effect of these antibiotics. So at that moment, we are saying that the bacteria is showing ampicillin resistance. Here we are going to stimulate a naturally occurring mutation and induce a genetic change in bacterial strains that make them ampicillin resistant. So first of all, we would insert some gene in the bacteria that would express beta lactamase, as we mentioned previously, which would allow this bacteria to grow, although the media contain ampicillin. The bacteria that we are going to use are Escherichia coli, and we are going to use another construct called plasmid. So what is plasmid? Actually, plasmid is a shuttle or a vehicle that's used to introduce external genetic material or DNA into the bacteria. It's a way for increasing the number of that require DNA. If it's a gene, it's a number to increase its expression. If I need to use that gene for forward injections, if I need to apply genome editing technology like CRISPR-Cas9 or TALENs or other zinc finger, there are lots of actually genetic engineering. I have to use a plasmid. Any one of you who are interested in using genome editing technology have to pass with molecular cloning and transformation. So first of all, the plasmid with ampicillin resistant gene, and then we insert the plasmid. Here is the Escherichia coli plus the plasmid with the ampicillin resistant gene. We would insert it inside the E. coli through a process called transformation, and we would cultivate that into ampicillin resistant bacterial culture. The objective is to use molecular biology tools, plasmid BUC18, to introduce new gene or sequence of an external DNA into the genome of Escherichia coli. You know that the bacteria have a circular DNA, and by introducing a foreign, what we can say, DNA into its genome, the DNA replication machinery would replicate the bacteria genome, which is the native one, and then it would also replicate the foreign DNA. You get the point? It's a kind of, we are infecting our bacteria or Escherichia coli with another external DNA, and we are relying on the DNA replication machinery to propagate our DNA. Thirdly, we would get rid of the bacterial DNA and only preserve our desired or external DNA. The steps, I would let you read it, and I think it would be better if we listen with each other to a tutorial about transformation. You can read the steps. I guess it wouldn't be a big issue, and I will provide you with several links, so it would be better for you to listen to it. I would run the tutorial now. It's a bacterial transformation, meaning introducing.
Transformation is the process by which foreign DNA is introduced into bacterial cells. Plasmid transformation is an important technique for microbiology experiments because it allows plasmids to be stored and replicated in bacteria. Scientists use genetic modifications and chemical treatments to create bacterial cells that are easy to transform. These are known as competent cells. In this video, we will show you how to use a standard transformation protocol to introduce plasmid DNA into competent cells in order to store and replicate your plasmid of interest. At the end of this video, we will talk about 2 alternative protocols that can significantly reduce transformation time or improve efficiency. To start the transformation process, thaw and aliquot a frozen chemical.
Here we begin the bacterial transformation. We would listen with each other.
Transformation is the process by which foreign DNA is introduced into bacterial cells. Plasma transformation is an important technique for microbiology experiments because it allows plasmids to be stored and replicated in bacteria. Scientists use genetic modifications and chemical treatments to create bacterial cells that are easy to transform. These are known as competent cells. In this video, we will show you how to use a standard transformation protocol to introduce plasmid DNA into competent cells in order to store and replicate your plasmid of interest. At the end of this video, we will talk about 2 alternative protocols that can significantly reduce transformation time or improve efficiency. To start the transformation process, thaw an aliquot of frozen chemically competent cells on ice for 30 minutes. There are several different types of chemically competent cells available commercially. You can also make your own using commercial kits or do-it-yourself protocols available online. Once acquired, competent cells are usually stored at negative 80 degrees Celsius. Multiple freeze-thaw cycles can reduce competent cell viability. To limit competent cell
So here we need to get only the vial that we are going to use. Don't thaw and freeze the cells, otherwise their cell wall would shrink and they would not be able to take any foreign DNA.
exposure to multiple freeze-thaw cycles, you should aliquot your cells into small volumes prior to storage and only thaw the amount required for your experiment directly prior to performing your transformation. As the competent cells thaw, place the appropriate LB agar plates on your bench. These plates should contain the antibiotic that your plasmid of interest will provide resistance to. The plates will reach room temperature by the time we're ready to spread our transformed cells. Next, add the plasmid DNA to the competent cells. Add between 10 picograms to 100 nanograms of plasmid DNA to your cells. Mix the cells and DNA by gently flicking the tube with your finger. We use DH5 alpha cells for most of our transformations because they are efficiently transformed by many plasmids. As a control, you should also mix a separate aliquot of competent cells with water and carry these through the rest of the transformation process. These cells should not grow once plated, but will give you confidence that your antibiotic works properly. Be sure to carefully label your samples and controls so you don't confuse them along the way. Next, heat shock the DNA competent cell mixture by placing the tube in a floating rack inside a 42 degrees Celsius water bath for 45 seconds. Then place the tube on ice for 2 minutes. This change in temperature increases the permeability of the bacterial cell membrane and allows the plasmid to enter the cells. After heat shock, we allow the transformed cells to grow for a short period of time in a shaking incubator. During this outgrowth step, the transformed bacteria are incubated in a rich medium like SOC, which contains glucose. This brief period gives the bacteria time to generate the antibiotic resistance proteins encoded in the plasmid backbone. This step is not as critical for plasmids containing ampicillin resistance and can be skipped in instances where ampicillin is used. To start the outgrowth step, first add 500 microliters of SOC to the tube containing the transformed bacterial cells. Next, place the tube in a shaking incubator at 37 degrees Celsius for 45 minutes. To increase the number of colonies you get after plating, you can concentrate your cells after the outgrowth step by centrifuging. We usually centrifuge the cells for 5 minutes at 4,000 RPM or 1,500 Gs.
Here we are centrifuging the whole aliquots and get rid of the excess media, and we only use below supernated and culture it on the media to increase the number of colonies rather than having solution or liquid on our agar, which increase the rate of infection or fungus growth. It's better to discard the media and use the deposit.
After you've removed some of the supernatant, you can resuspend the cells and pipette them on top of a warmed LB agar plate containing the necessary antibiotic. If you use pipette tips that are easy to bend, like ours, you can also use them as spreaders when plating the cells. Be careful when bending the tip and make sure you are using good sterile technique throughout the process. The last step is to place the agar plate into an incubator for overnight growth. While most strains require incubation at 37 degrees Celsius, some may require lower incubation temperatures, such as 30 degrees Celsius. After overnight incubation, inspect your plates for visible colony growth. Remember, your control plate should have no visible colonies present. If your sample plate has colonies that are too close together, you can pick a clump of cells and streak for single colonies on a new plate.
Here it said that the control should not have any cross, because actually the control here only is the original E. coli plate on the plate without any plasmid. It should, all of them are dry because the plate contain ampicillin. In case if there is a cross, this means that the efficiency of ampicillin is not good, means it allows the cross of bacteria and other fungus.
See our Streaking for Single Colonies video for more information. Although this protocol will work for most plasmids, it can be shortened for easy to transform plasmids or altered for plasmids that are difficult to transform. If you want to significantly reduce the time needed for transformation, skip the heat shock and outgrowth steps entirely and simply incubate the DNA competent cell mixture on ice for 5 minutes before plating. This could impact your transformation efficiency, but will work for samples that are easy to transform. You might not always get transformants the first time you try. This could be due to the reagents you use or the size of the construct you are transforming. You should check that your competent cells are still viable and that you use the right antibiotic. If your construct is larger than 10 kilobases, it is a good idea to use electric competent cells and an electroporator instead of heat shock. Unlike chemical transformation, which relies on heat shock to cause the cells to take up DNA, electroporation uses an electric field to induce membrane permeability and often leads to higher transformation efficiency. Thank you for watching Adgene's transformation protocol video. Check.
Okay, so this is the point that we are talking about. Uh, how to introduce the DNA, it's either through chemical, which is known as transformation. Both of them are transformation, either chemically, relying on the heat shock, meaning that I would use heat shock by applying high temperature for 40 seconds and followed by low temperature through keeping the cells in the ice, which would change the permeability of the cell wall of the bacteria. At that point, the foreign plasmid or the foreign vector carrying our DNA or gene of interest would be able to enter inside the bacteria, and then the bacteria would heal itself and close its cell wall. Permeability of the cell wall would be would return to the normal. Afterward, when we cultivate these bacteria on the suitable agar with the antibiotic resistant, it would be able, the DNA machinery, replication machinery would replicate both the bacterial DNA and the plasmid DNA. Afterward, we would isolate only the bacterial DNA and get the bacterial DNA and plasmid DNA and get rid of the bacterial DNA. Okay. So you can read those steps. They are not hard. So contain plasmid by letting them grow on ampicillin containing medium, because the plasmid here contains a gene expressing beta lactamase that would destroy the ampicillin beta lactam ring. And here, as we mentioned, we put them in a bucket with ice. Then we keep them at a high temperature for 40 seconds, 42 degree water bath. It may be by maximum 40 seconds or two, not more than 40 seconds. Then put them in ice. This is the process of the heat shock, changing the temperature from cold to very hot to cold again, so the cell permeability would change, allowing the foreign DNA to be entered. Then we put them again into the rack to reach ambient temperature, and we would keep them on a shaker to allow the mixing of the bacteria with the foreign DNA in the suitable media. It's called actually LB media. Gloria Pross, she is a scientist who invented this media. And then we would centrifuge that and take 200 microliter and cultivate that on the suitable media. As we mentioned, we can skip the process of heat shock, but this would not be, it wouldn't give a good results. Okay. I think next week we can set meeting to discuss anything which is not clear for you. Also, I'm giving you my contact number on Friday. You can contact me at any time between 9 to 12 and 1 to 4 p.m. I would also provide you with tutorials. You have to listen to it carefully to understand if anything is missed. Also, Dr. Alice would upload the homeworks. Make sure that you you submitted your homework before the deadline. Okay. At the end, I'm so thankful for your kind attention, hoping that everyone is fine and safe. Hope to see you soon. Maybe by the next semester, everything would be better. Stay safe. Take care of yourself. Bye bye. Salam.